Hello everyone, welcome to CBRC University Biology Module number 3. So the title of our module is the cellular energy. For the overview, in here we're going to tackle the introduction to cellular energy, the difference between autotrophs and heterotrophs, the energy storage and release mechanism of different organisms, the energy exchange, the process of photosynthesis, which is subdivided into different stages, the light-dependent and the light-independent reaction, the cellular respiration, as well as the processes such as glycolysis, Krebs cycle, and the electron transport chain, the alcoholic fermentation, and of course, the last, the lactic acid fermentation. So to start, let's go from the basic. So the term energy was used since we were in elementary. So when we say energy, this is the ability or the capacity to do work. To sell, to a cell, work includes the metabolism processes such as the growth, the repair, the digestion of food, the process of phagocytosis and endocytosis, like what we discussed in our module number 2, all about the biochemistry. So the main difference between autotrophs and heterotrophs. When we say autotrophs, the term or the word trophs literally means nutrition. Autotrophs meaning they are creating or producing their own food or nutrition. So when we say autotrophs, these are the organisms that can use the energy of sunlight, carbon dioxide, and water to make their own food. So the other term for autotrophs are producers. Examples are plants, since they are photosynthetic organism. We have algae, since some of algae has the chlorophyll or known under the group of chlorophyta, and also some photosynthetic bacteria like the blue-green, the cyanobacteria. Next is the heterotrophs. When you say heterotrophs, hetero meaning other. So they derive their nutrition or food from the others. So these are the organisms that cannot produce their own food. So they are also known as the consumers. So the word consumer meaning they consume another organism. So examples are animals, of course, the fungi, and other bacteria. How about the energy storage and release? When we say energy storage, that is the mechanism or the ability of an organism to store energy in different form. And release is the ability of the organism to metabolize the larger molecules into smaller molecules to be used as a form of energy. So in all or some of the organism uses this nucleotide called ATP. When we say ATP, that is the adenosine triphosphate. So that is the main energy storage molecule by most organisms. And as what you've seen in our slide, ATP is composed of three major parts. We have the ribose, the adenine, and the phosphate. So ribose, of course, is a 5-carbon sugar molecule. We have the adenine, that is one of the nucleotides of RNA and DNA. And of course, the phosphate, which serves as the backbone of the ATP molecule. How about the energy exchange? So energy exchange is composed of mainly two separate processes. We have the reduction and the oxidation. When we say reduction, that is the addition of electrons to an atom or an ion. So it is quite confusing because reduction is addition. So always remember that reduction is addition of electron. It is termed or called as reduction because when you add an electron to a certain atom, you reduce the charge of the atom. So the term or the title or the name reduction was derived due to the effect of adding electrons. While oxidation is the losing of electrons. So when we say reduction, that is the adding, while oxidation is the losing of an electron from an ion or an atom. But when you combine reduction and oxidation, we will have this what we call redox reaction or the reduction oxidation reaction. So this is a chemical reaction which one molecule is oxidized and the other molecule is reduced. Okay, so how about the cellular energy processes? So the main processes involves uh, in the use of different energy or larger molecules in an organism is called metabolism. And metabolism is divided into two main processes. We have the catabolism and the anabolism. When we say catabolism, that is the energy-yielding metabolism, while the anabolism is the biosynthetic metabolism. When we say catabolism, energy-yielding, meaning that is the process or that is the metabolism in which you derive energy from larger molecules. So, in simpler explanation, catabolism is simply the derivation of energy from the biomolecules, while anabolism is the production of biomolecules for storage. So the difference of the two is that catabolism is to extract energy, while anabolism is to preserve energy in forms of biomolecules. And the main cellular energy processes occurring in some organisms but not all are the photosynthesis and the cellular respiration. In plants, of course, photosynthesis and cellular respirations occur. But in animals, photosynthesis does not occur because they don't or we don't have this organelle called the chloroplast. So let's start with the photosynthesis. So the etymology of photosynthesis is the word photo, of course, it means light, and synthesis means putting together. So it means that it is the process used by autotrophs in which they convert sunlight or light energy into a usable form of energy. So for the chemical formula or equation for the photosynthesis, we have six molecules of carbon dioxide added to six molecules of water. So using the sunlight or the energy coming from the sun, we can yield one molecule of glucose or C6H12O6 added to or together with six molecules of oxygen as the byproduct. So, in the next slide, I have here the picture of the sun. It is because the sun is the main source of energy in our planet Earth. It is the energy from the sun travels through the space in form of sunlight. And of course, you all know that sunlight it can be a particle or a wave. So, a particle, the particle of sunlight is called photons, and also it travels in a wave with different wavelengths. So the energy of the color of the light is directly proportional to its wavelength. Okay, so how plants collect energy or how plants collect photons from the sunlight? So that is through the help of light trapping pigments. So when we say pigments, these are the molecules or substances that can absorb light. So as what I've said earlier, photons are the packets of light or those are the particles of light energy that travels as a wave. And photons have different wavelengths. So in the diagram or picture provided in the slide, we can observe that the color or the color of the light with the longest wavelength is the red. So meaning, the longer the wavelength, the lower the amount of energy. While the violet has the shorter wavelength. So the shorter the wavelength, the higher the amount of energy. How about the types of chlorophyll? So again, chlorophyll is the green pigment found in green plants. So chlorophyll is also a form of pigment which is responsible in absorbing the photons or light energy. So there are two types of chlorophyll, the chlorophyll A and the chlorophyll B. So they have the same function but they differ in the intensity of their work. So when we say chlorophyll A, it has the main primary or it has the primary function or it has the primary contribution in the absorption of light. While chlorophyll B has a slight contribution or it serves as a backup plan for the chlorophyll A. So if chlorophyll is the green pigment, how about the other color? So we have the carotenoids or carotenoids. When we say carotenoids, it includes the yellow, the red, and the orange pigment, which is responsible in giving colors for carrots, bananas, squash, and dried leaves. So in the figure, you have the green the green colored leaves of maple trees. So during fall season, the carotenoids will spread throughout the leaves. So it will turn the leaves into reddish orange color. So where does the photosynthesis reaction takes place? So of course, the photosynthesis happens in the chloroplast. So what is chloroplast? Chloroplast is the organelle that contains the chlorophyll and it is where the photosynthesis occurs. So what are the parts of chloroplast? We have the three main parts of chloroplast. We have the stroma or the liquid substance inside the chloroplast. We have the thylakoids or the flattened channels and discs. And of course, we have the grana or the stacks of thylakoids. So thylakoids are the discs, and when you put them together in stacks, so they are now called as grana or granum. So I have here the diagram or illustration of the parts of the chloroplast. So again, so chloroplast is a double layered membrane. So we have the outer membrane, the inner membrane. We have the granum or the stack of thylakoids. We have the thylakoid, the stroma, and the intermembrane space. How about stomata? So stomata is the tiny opening or pore that is used for gas exchange. So thylakoids found in the chloroplast contains the chlorophyll, which is responsible in collecting sunlight or the light energy, while stomata serves as the site of gas exchange. It is because carbon dioxide or CO2 is very important that serves as a raw material for the photosynthesis. And of course, the stomata is found mostly under surface of the plant leaves, especially in the spongy layer, while the chloroplast is found in the palisade layer of the leaf. So it is composed of the palisade layer, the mesophyll, and the spongy layer. So chloroplast at the top of the leaf and stomata at the bottom of the leaf. Okay, so I do an illustration, a summary illustration of the reactions in the photosynthesis. So, as you can observe, we have the light-dependent reaction and the light-independent reaction. So, we're going to discuss the different processes and the difference of the two. So, let's start with the light-dependent reaction. So, the term itself suggests that light-dependent reaction, meaning that this type of reaction depends primarily on the use or in the presence of sunlight. So, light-dependent reaction, it is the first reaction. Take note, that is the first reaction in the photosynthesis. It is the process in which light energy is converted into chemical energy of ATP. And take note that the light-dependent reactions occur in the thylakoid membrane. Again, the light-dependent reaction occurs in the thylakoid membrane. So what are the raw materials for the light-dependent reaction? We have the light energy and the water. So light energy to be gathered by the chloroplast with the help of the photosystem or the light collecting system of the plants, and water which is absorbed by the roots of the plant. So the light-dependent reaction is composed of four main stages. We have the chlorophyll photoactivation, we have the photolysis, the ATP formation, and the NADPH formation. Again, we have the four main stages. The chlorophyll photoactivation, the photolysis, the ATP formation, and the NADH formation. So let's start with the chlorophyll photoactivation. So when we say chlorophyll photoactivation, of course, the main actor in this stage is the chlorophyll. So since chlorophyll, we have the two types of system or the collecting system. We have the photosystem 1 or the P680 and the photosystem 2 or the P700. So in this photosystem, they are responsible in collecting. They are within the chloroplast and responsible in collecting the photons. So when this photosystem receive the photons, they excite or they change the charge of the electrons from ground state into excited state. And then this excited state or this electron travels outside the photosystem or the outside the light collecting system or the photosystem itself. And of course, in the process of chlorophyll photoactivation, we also have the photolysis. So photolysis meaning with the use of sunlight, okay, with the use of sunlight, we can separate the molecules of water. As what you can remember, water is a raw material in the light-dependent reaction, right? So for us to use water, the water must undergo this process called the photolysis. In the use of sunlight, we're going to separate the molecules of water. So again, the formula for water is H2O. So we're going to separate the hydrogen ions to oxygen molecules. So the oxygen molecules will be produced or will be released outside the environment. Just like the illustration, the O2 molecules was released outside the environment, while the hydrogen molecules will be further used in the next process. And also, when you separate the hydrogen and oxygen from water, you release two uh, electron molecules. And these electron molecules will be excited once again in the chlorophyll photoactivation. And these excited molecules will be transferred into the other processes. So these processes are the ATP formation and the NADPH formation. So ADP or adenosine diphosphate and NAD or the nicotinamide adenosine are both present inside the chloroplast. So the process of formation is that the hydrogen ions or the hydrogen ions derived from the photolysis will attach themselves to the ADP molecules, producing ATP molecules, and the hydrogen ions will attach themselves into the NADP, producing NADPH molecules. So now, the products of light-dependent reaction is the oxygen, which is released into the environment, and the ATP, which is a form of energy, and the NADPH, which is a high-energy electron carrier. So, in the next process, which is light-independent reaction, meaning independent, we don't need the light anymore. So, we need the ATP produced from the light-dependent reaction, and of course, the high-energy electron carrier, the NADPH. Okay, to sum up the light-dependent reaction, we have here the formula. So the light energy, which is derived from the chlorophyll through the use of chlorophyll in the process of chlorophyll photoactivation, in which they excite the electron that travel from one photosystem to another, and of course the water, which is collected or absorbed by the roots of the plant, which is further separated into its component, the hydrogen and oxygen, which is used by which the oxygen is released through the environment, and the hydrogen is added to the ADP, forming ATP, and the NADP forming NADPH. Again, ATP is the storage molecule for energy, and NADPH is the high energy electron carrier. So again, the byproduct of this process is the oxygen. Since oxygen is released to the environment and used by other organisms like humans and animals. Okay, so we will now proceed to the light-independent reaction. So light-independent reaction meaning it is now independent. So this process doesn't need the light anymore. So this process uses the products of light-dependent reaction to convert carbon dioxide and water to sugar. So again, the products of the light-dependent reaction are the ATP and the NADPH molecules, which is further added to the carbon dioxide, which is derived from the environment through the help of the pores under the leaf, which is called the stomata. So again, the raw materials are the ATP, the NADPH, and the carbon dioxide. So light-independent reaction is composed of three main stages. We have the carbon dioxide fixation, the reduction, and the regeneration. So I have here the light-independent reaction. As what you observe, the diagram or the illustration is in cyclic form. It is because light-independent reaction is also called as the dark reaction. Dark because it doesn't need light, and it is also called as Calvin cycle in honor of Melvin Calvin, who discovered this process. So it is a cyclic form because there is a certain substance that is recycled throughout the process. So again, the three main stages of the light-independent reaction are the carbon dioxide fixation, the reduction, and the regeneration. So let's start with the stage 1. So stage 1 is the carbon dioxide fixation. So of course, again, carbon dioxide is derived from the environment through the help or in the gas exchange site, which is the stomata. So the carbon dioxide will now enter the Calvin cycle, in which it combines with the 4 carbon molecule. So in the process of carbon fixation, the carbon dioxide, which combines with the 4 carbon molecule, will be divided into two molecules of 3-PGA, or the glyceraldehyde phosphate. These molecules of glyceraldehyde phosphate will be submitted into the stage number 2. The second stage is called the reduction. It is called reduction because the every molecule of glycyl aldehyde phosphate will be reduced into 6 molecules of NADPH or nicotinamide adenine phosphate. So these molecules of NADPH carries a lot of high energy electrons, which produces one molecule of 3-PGA or the 3-PGAL. So these molecules are needed in the production of glucose. So this cycle is very complicated because the Calvin cycle must rotate 6 times in order to produce one molecule of glucose. So again, we have the carbon fixation followed by the reduction in the process of reduction, the molecules of PGA or the or the glycyl aldehyde phosphate is reduced to NADPH. So the third step is the regeneration. The third step is called regeneration because it is the stage in which a certain substance is regenerated, and that substance is called the rubisco. So rubisco or the RUBP is regenerated. So RUBP is an important enzyme used in the carbon fixation for us to convert carbon dioxide into PGA molecules. So again, the rubisco is regenerated and further used in the carbon fixation once again. So as what I've said earlier, it is a cyclic process. The rubisco is recycled again and again because it is used in the carbon fixation. So again, Calvin cycle must rotate 6 times in order to provide one molecule of glucose. So the formula for glucose is C6H12O6. So to summarize the formula for the light-independent reaction, we have the ATP molecule plus the NADPH carrier plus the carbon dioxide yield C6H12O6 or called the glucose. So for us to differentiate the light and the dark reaction, so light reaction is the light dependent and the dark reaction is the light independent reaction. So for us to differentiate the two, when we say light reaction, of course, the light is needed to produce organic energy molecules such as the ATP and the NADPH. So ATP and the NADPH are the products of light reaction, which is further used in the next process called the dark reaction, also called as the Calvin cycle, also called as the light independent reaction. So in dark reaction, no light energy is needed. Instead, dark reactions are use ATP and the NADPH molecules produced from the light dependent reaction to produce this carbohydrate, also called as the glucose molecules. So for the general equation, again, we have the 6 molecules of carbon dioxide used in the light-independent reaction added to the water molecules used in the light-dependent reaction in the process of photolysis yields through the use of light energy produced one molecule of glucose and of course the byproduct which is the oxygen. So again, carbon dioxide plus water yields through the use of light energy produces glucose and oxygen. So that is the general equation of the process of photosynthesis. How about the importance of photosynthesis? So, number one, glucose, since sugar, as what we discussed in the biochemistry, carbohydrate provides energy for the organism. And glucose is a form of carbohydrates. So, glucose provides the basic energy, or it serves as the basic energy source for all living organisms. So, in the illustration or picture, we have here the different kinds of vegetables and fruits. So, we all know that fruit and vegetables are good source of glucose because they are products of plants. So, through the process of digestion, animals can derive energy or they can derive glucose from those foods and materials. Next importance is that oxygen is a vital source for respiration of all living organisms. Of course, as humans, animals, oxygen, the byproduct of photosynthesis, is very important because oxygen is food of the brain. So, and also oxygen is needed in our cellular metabolism and processes. That's why in the process of respiration, which is further discussed in the human anatomy and physiology, oxygen was absorbed in the gas exchange site in the alveoli of the lungs and transferred with the help of hemoglobin, transferring the the oxygenated blood throughout the body. So that is important in all living organisms like humans. And the third one, the last one, is that the photosynthesis is the ultimate source of energy that drives all metabolic processes in nature. So as what I said earlier, glucose, which is the product of plants, which is stored as a form of polysaccharide molecule called the starch. So starch is the polysaccharide molecule that serves as the storage of energy of plants. So photosynthesis is the ultimate source. Take note, the ultimate source of energy since plants provide the energy, or since the autotrophs was consumed by the heterotrophs, which is further used for the energy metabolism, or in the production of energy for further work or other processes to support the daily living of different cells. So again, those are the importance of photosynthesis in the different organisms and of course in our nature. So again, the cellular energy processes are the photosynthesis and the cellular respiration. So photosynthesis is a form of anabolism. Again, when we say anabolism, that is the production or creation or building up of biomolecules to store energy. So in the photosynthesis, to store energy, they store that in form of starch or the polysaccharide molecule. While the other process called the cellular respiration, which is a form of catabolism, when we say catabolism, that is the breaking down of molecules to produce energy. Cellular respiration is the opposite of photosynthesis. Why? Because in cellular respiration, glucose serves as the raw materials and it will be break down to produce ATP, which is the opposite process of photosynthesis. So again, what is cellular respiration? Cellular respiration is the process by which food molecules are broken down to release energy for work. It appears to be the reverse photosynthesis, and because of the products of photosynthesis serves as the raw material. So let's observe the illustration or the general equation of the two. So in photosynthesis, the raw materials are carbon dioxide and water, while in respiration, carbon dioxide and water are the product of respiration. In photosynthesis, glucose molecules and oxygen are the product, but in respiration, glucose and oxygen are the raw materials. So meaning really that these two processes are opposite in nature. So, if photosynthesis occurs in the chloroplast, where does the cellular respiration takes place? So, the cellular respiration takes place in the mitochondria. That's why the mitochondria gained its title as the powerhouse of the cell. It is because the term powerhouse, meaning it produces or it provides energy for the cell. So, mitochondria, it is a double membrane like the chloroplast. It is a double membrane, the inner layer with folded inwards called the cristae, which is the site of cellular respiration. So in the diagram, in the slide, we have the outer mitochondrial membrane, the inner mitochondrial membrane, we have the cristae and the matrix, or the mitochondrial matrix. So, cellular respiration is composed of three stages: the glycolysis, the Krebs cycle, and the electron transport chain. However, other books suggest that cellular respiration is composed of four stages. We have the transition stage between glycolysis and Krebs cycle, but I include that in the presentation. So again, the three main stages of cellular respiration are the glycolysis, the Krebs cycle, and the electron transport chain. So let's start with the glycolysis. When we say glycolysis, it is the process by which glucose is converted to pyruvates, in which the energy is released. And take note, glycolysis occurs in the cytoplasm of the cell. So the term suggests that glyco means carbohydrates, and lysis means splitting or the separation or splitting of molecules. So glycolysis is the splitting of glucose molecules. So, in the separation of glucose molecules, it produces ATP or energy. And of course, the separation of glucose molecules, they produce two molecules of pyruvate. So, I have here the illustration. So, glycolysis, we have the preparatory stage. Preparatory stage because the glucose must be prepared first. So in this stage, the 6-carbon molecule of glucose will be separated into two. So again, take note, the glycolysis is composed of two main processes, the energy investment phase and the energy harvesting phase. So energy investment, meaning in the process of glycolysis, we have to invest two molecules of ATP. Okay, so in the diagram, you can observe the two yellow molecules, so it is colored yellow. The two molecules of ATP was invested to the process. So the one molecule of glucose with the help of ATP molecule was reduced into pyruvate molecule. But since glucose is a very stable molecule, it cannot be separated easily. That's why we have to invest another ATP molecule for us to convert the glucose molecule into pyruvate or the fructose molecule. And these pyruvates will be used in the next stage. So again, we invested two ATP molecules. But in the process of glycolysis, we can produce four molecules of ATP in every division. But since we invested two molecules of ATP and we harvested four molecules of ATP, totally the net gain of ATP is two molecules since we invest two and we harvest four. So if we're going to compute that mathematically, so 4 minus 2, so the gained 4 electrons minus the invested 2 ATP, meaning we only gained 2 ATP molecules. So for the glycolysis process or the glycolysis stage, we have 2 ATP gained molecules. So these pyruvates will be used in the process of Krebs cycle. But take note, pyruvates cannot be used in Krebs cycle. That's why we have this what we call transition stage. So in this process, transition stage, pyruvates will be converted into another form of molecule. So let's see what will happen. So pyruvate is the product of glycolysis. So since pyruvate cannot be used directly in the process of Krebs cycle, so from the cytoplasm, the site of glycolysis, the pyruvate will travel to the transport protein within the mitochondrion. So the pyruvate will combine to coenzyme A or CoA, producing acetyl coenzyme. The combination of these two, the pyruvate and the coenzyme A producing acetyl coenzyme, produces carbon dioxide molecule, which is a product of cellular respiration. And of course, the combination of of coenzyme A and pyruvates producing acetyl coenzyme produces NADH, which are high energy electrons carrier. So now we have the acetyl CoA, which is the product of the transition stage. This acetyl CoA will be used in the next stage, which is the Krebs cycle. Uh, the name Krebs cycle was honored after Hans Krebs. Okay, so when you say Krebs cycle, it is also called as the citric acid cycle because in this stage, we're going to produce the citric acid. And also, it is a series of oxidation reactions. Again, oxidation reaction is the loss of electron that makes up the second phase of respiration. So to summarize the process of Krebs cycle, I have here the diagram of the Krebs cycle. So again, so the glycolysis, we already produced the pyruvate. So this pyruvate was converted into acetyl coenzyme as it combines with the coenzyme A in the transition stage. So now we have the acetyl coenzyme. It will now enter in the process of Krebs cycle. So this acetyl coenzyme will combine with a certain substance, a 4-carbon substance, producing the citric acid. That's why Krebs cycle is also called as the citric acid cycle. So this citric acid cycle undergoes series of oxidation reaction. So again, oxidation reaction meaning electrons was released in the environment. And of course, the hydrogen molecules was also released to the environment as well as the carbon dioxide. So these hydrogen molecules was further picked up by the NADH, producing the NADPH molecules. So it is called a citric acid cycle because the citric acid cycle was later submitted into different oxidation reactions. So it is converted into isocitrate, ketoglutarate, succinate, uh, and so on and so forth. So meaning in this process, it is a recycling process because the four carbon molecules called the oxytoxate or the oxyacetate, oxaloacetate, will be further used again in the first part in which it combines with the acetyl coenzyme, which is further submitted to produce the citric acid cycle, which is further submitted again into oxidation reaction. So meaning it is a cyclic process. But take note that in the process of Krebs cycle, the main purpose of the Krebs cycle is to produce the two main molecule, the FADH and the NADH. When we say FADH, that is the flavine adenosine diphosphate, or the flavine adenosine dinucleotide. So when we say NADH, that is the nicotinamide adenine dinucleotide. So those two molecules are important for the next process. So again, the products of the citric acid are the FADH and the NADH, which is the raw materials for the next process. So the next process is called the electron transfer chain, which is also called as the oxidative phosphorylation. So what is electron transport chain? So electron transport chain is the movement of electrons from NADH and FADH in the inner membrane of the mitochondrion. The movement of electron inside the mitochondrion results in the formation of the ATP molecules. So I have here the diagram of the electron transport chain. So again, the the main raw materials for the electron transport chain are the products of the Krebs cycle, which are the NADH and the FADH. The nicotinamide adenosine dinucleotide and the flavine adenosine dinucleotide. So it will further be used in the transport chain. How? So the NADH. So since the NADH contains, they serve as high energy electrons carrier, the electrons will be released into the inner membrane of the mitochondria, as well as the molecule carried or the electron carried by the FADH molecule, releasing hydrogen ions in the mitochondria. So these hydrogen ions will now travel outside the inner mitochondrial membrane. They will pass through the inner mitochondrial membrane. So now, when the hydrogen passes from inner to the outer membrane, they created the concentration gradient, meaning there are more hydrogen outside than inside. So what will happen? So hydrogen will now travel from higher concentration into the lower concentration. So in the process of in the process of transfer from higher concentration to lower concentration, the hydrogen ion will pass through a certain protein. This protein is called the ATP synthase. The ATP synthase is the protein responsible in the production of ATP. Why? Because when a hydrogen ion passes through the ATP synthase to maintain the gradient. So to maintain the gradient, they produce or they combine with the ADP molecule. So ADP molecule again is a molecule which is already present in the membrane. So the high energy electron combined with the ADP molecule producing the ATP or the adenosine triphosphate. So that is through the help of the ATP synthase. So it is a chain process because as the process goes on and on, the hydrogen molecules creating the concentration gradient travels from inside the mitochondrial membrane, outside the mitochondrial membrane, passes through the ATP synthase, forming the ATP. So again, this ATP will now be used by the cell. But now, another NAD and FADH molecule will now travel into the mitochondrial membrane, releasing the hydrogen molecules which travel again outside the cell, forming the concentration gradient. So again, this is a cyclic and chain reaction. So I have here the diagram or the summary of the electron transport chain. So the cellular respiration. So in the glycolysis, we produce two molecules of ATP. In citric acid, we produce another molecules of or another two molecules of ATP. While in the oxidative phosphorylation or the electron transfer chain, we produce 34 molecules of ATP. Again, two molecules are produced in glycolysis. Two molecules are produced in citric acid and 34 molecules are produced in the electron transfer chain. So the total is we have 38 ATP molecules produced in the process of cellular respiration. So to sum up the process of cellular respiration, in the process of glycolysis, again, the glucose was converted into pyruvate, releasing two molecules of ATP. So again, so since pyruvate can no longer be used directly in the Krebs cycle, it is first to be converted in the transition stage or the formation acetyl coenzyme with the help of the coenzyme A. So now we have the acetyl coenzyme. It will be used in the Krebs cycle or the citric acid cycle. So this acetyl coenzyme will bond with the oxaloacetate, which is further submitted into different or into different oxidation reaction, which is cyclic in nature, producing two molecules of ATP. And again, so the main product of Krebs cycle are the FADH and the NADH, which are high energy electrons carrier, which is used in the electron transport process, producing 32 molecules of ATP. Again, in the NADH and the FADH releases their electrons, in which these electrons or hydrogen ions travels from the inner membrane to the outside of the membrane, creating the concentration gradient. So when the concentration gradient was provided, the hydrogen molecules will now travel in the ATP synthase, forming the adenosine triphosphate. Since in the process of electron transfer chain, it produces 32, or some book says that it is 32 ATP, but the other book says that it is 34 ATP. So again, the general formula for the cellular respiration is the glucose molecule added to the oxygen molecule added to the water molecule yields or produces carbon dioxide as the byproduct, and of course, water and the ATP, the energy, which is the end product of the cellular respiration. So energy will be further be used in the other metabolic or in the cellular processes of the body of the different organism, plants or animals. So there are two general classifications of respiration. So it's either aerobic or anaerobic. So the general difference is that aerobic respiration, it requires oxygen, while the anaerobic respiration, it does not require oxygen. So let's differentiate the two. In terms of oxygen requirement, as what I've said earlier, aerobic does not always or does not need any oxygen, but anaerobic, never. They don't need oxygen. Waste product. So in aerobic respiration, carbon dioxide and water are the products, while in the anaerobic respiration, carbon dioxide and ethanol, an alcohol, take note, ethanol or alcohol is the product. In the efficiency, aerobic respiration is very efficient, while in the anaerobic, it is less efficient. So in the terms of energy release as heat, in aerobic respiration, of course, some energy was released as heat, while the anaerobic respiration, some of the molecules was not released as heat. So let's proceed to the examples of anaerobic respiration. So when we say anaerobic respiration, it is a chemical reaction that releases energy from food molecules in the absence of oxygen. So absence of oxygen. So it can be alcoholic fermentation or the lactic acid fermentation. When we say alcoholic fermentation, in the absence of oxygen, the unicellular fungus, also called as the yeast, converts pyruvates into carbon dioxide molecule and ethanol. So the formula is pyruvates from plus the NADH converted through the help of yeast producing ethanol, NAD molecules, and the carbon dioxide. So alcoholic fermentation is very popular in winemaking, process, and also for the bakers. Why bakers? Because uh, the the yeast is the responsible making in making the bread rise. So in the diagram provided, as you can see, in the process of alcoholic fermentation, the bubbles which contains carbon dioxide produced by the yeast was inside the bread. But when the bread was baked, these spaces or the gaps or the spaces in the bread was due to the fact that there are bubbles inside there, the bubbles of carbon dioxide. And also, aside from carbon dioxide, alcohol is also a product of the alcohol fermentation. So, where's the alcohol in the bread? Since bread is submitted into intense heat inside the oven, alcohol already evaporated. That's why you can only see the spaces, which are the spaces of the bubbles of carbon dioxide produced by the yeast. So I have here the diagram of the alcohol fermentation. So the glucose molecule again was submitted. It was divided into pyruvates, releasing two molecules of ATP and producing carbon dioxide molecules. These carbon dioxide molecules are stored in the bubbles or as bubbles inside the bread. So providing the spaces, making the bread rise. And of course, again, the alcohol can no longer be found inside the bread because it already evaporated. And sugar, it remains in the bread as a form of carbohydrate, which is further be metabolized by the consumers of the bread. How about the other one? How about the lactic acid fermentation? So the difference of the two is that in the alcohol fermentation, the product, of course, is the ethanol or alcohol. While the lactic acid fermentation, the product is an acid, which is a lactic acid without the formation of carbon dioxide. So an example situation is that when the muscles run out of oxygen, again, it is anaerobic, the cells switch from aerobic respiration to lactic acid fermentation. The process transfers hydrogen to pyruvates, converting the pyruvates into lactic acid, oxidizing the NADH and the NAD molecules. The best example for that is when you suffers from muscle fatigue or sprain. It is due to the fact that our muscles converts glucose into energy, right? How about through the process of aerobic respiration? How about if there's no longer oxygen in the body or in the cell? So the cell will now convert from aerobic respiration to anaerobic respiration in the form of lactic acid fermentation. So in this process, the glucose was again divided in the help of glycolysis into pyruvic molecules or pyruvic acid. So this pyruvic acid will be regenerated through the help of NADH molecules producing the lactic acid. So that's why you suffers the pain or when you work extraneously or when you work too hard in the gym, when you lift heavy materials, you suffer when you suffer muscle pains or muscular pains, muscular fatigue, or muscular sprains. So that is due to the fact that lactic acid was deposited or present in the muscle. Is since acid is very potent or high potent or acidic in nature, or as I can say that it is very it contains irritating molecules. Okay, irritating molecules to the cell. It provides the pain feeling or the uncomfortable feeling for our body. That's why when you work too hard, when you work extraneously, lactic acid was produced in our body, causes muscle fatigue or muscular pain. So as a summary, the photosynthesis is composed of the light reaction and the dark reaction. So the light reaction converts light energy or the light energy into usable form. So again, the products of light reaction, the ATP and the NADPH, will be further be used in the process of Calvin cycle, producing the sugar molecules or the glucose. While the cellular respiration, these glucose molecules will be divided in the process of glycolysis, producing the pyruvates, while this pyruvate will be converted into acetyl coenzyme with the help of coenzyme A, which is to be used in the Krebs cycle, submitted into oxidation reaction, producing the NADH and the FADH molecules, which is further be used in the electron transfer chain, producing 34 or 32 molecules. So 32 molecules, 32 to 34 molecules of ATP. So 2 molecules for glycolysis, 2 molecules for Krebs cycle, and 34 molecules for the electron transport chain. So those are the processes of cellular metabolism. So for the general equation, so six molecules of carbon dioxide added to six molecules of water added to the sunlight energy produces one molecule of glucose and uh, oxygen as the byproduct. While for the respiration, one molecule of glucose added to the six molecules of oxygen yields six molecules of carbon dioxide released to the environment and produces six molecules of water and energy, which is used for the daily living of the cell. And of course, with the cellular fermentation, the glucose was converted into ethanol, or it can be lactic acid, producing carbon dioxide and energy. So as you can observe, the photosynthesis and cellular respiration are opposite. Why? Because the glucose is a product of photosynthesis, while it is a raw material in the cellular respiration. So it's trivia time. So for our trivia, did you know that there is an animal that undergoes the process of photosynthesis? So meaning it is now a myth that all animals undergoes the process of cellular respiration, while only plants undergoes the process of photosynthesis. This animal is named the Elysia chloretica. So Elysia chloretica is a sea slug appearing as a giant swimming leaf. So of course, it is an animal which appears like a leaf. So it was recently found that it is the first known animal that undergoes the process of photosynthesis. How is it possible? It is because this sea slug consumes algae, right? And we all know that algae are photosynthetic materials. In the process of evolution and adaptation, this sea slug consumes the 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 photosystems of algae, making it in its own system, making that sea slug as a photosynthetic organism. Now, this Elysia chlorotica was recognized as the first organism that undergoes photosynthesis. And to check whether you learned something from our module, again, we're going to have our brain shaker. Direction, you have to identify the words or terms being defined or described in each item. So you can answer orally or you can write it down. So are you ready? Let's start. So for item number 1, it is called as the ability to do work. So it is the center of our discussion or of this module. It is the ability to do work. Correct. The ability to do work is called the energy. How about this one? The organisms that are capable of using energy to form sunlight to make their own food. Okay, so these organisms are called autotrophs. Very good. So autotrophs are the organisms that are capable of using energy from sunlight to make their own food. How about this one? A reaction that involves the addition of electrons. So it is one of the energy exchange reactions. So a reaction that involves addition of electrons. So correct. That is the process called reduction. So that is the opposite of oxidation. So in the process of oxidation, an atom loses an electron. Very good. How about this one? It is the process by which autotrophs convert sunlight into usable form of energy. So this process is also a form of anabolism because it provides or it produces glucose. What do you call to this process? Correct. This process is called photosynthesis, which is the opposite of cellular respiration. How about this one? It is the organelle of plants that contains photosynthetic pigment. The organelle of the plants that contains photosynthetic pigment. What is your answer? Okay, correct. That organelle is called the chloroplast, which contains the chlorophyll inside the thylakoid and composed of different parts, the grana, the stroma, and a double layer membrane. Next, What do you call to the light collecting unit of the chloroplast? I mentioned that earlier. We have two types. We have two types of the light collecting unit of the chloroplast, which contains the chlorophyll. Correct. The light collecting unit of the chloroplast is called the photosystem. So in the chloroplast, you have the photosystem 1 and the photosystem 2, which is responsible in the chlorophyll photoactivation. Next. It is the process by which glucose is converted into pyruvate. What do you call to the process by which glucose is converted to the pyruvate? It is the first step of cellular respiration and occurs in the cytoplasm. Correct. This process is called glycolysis, the process by which glucose is converted into pyruvate, the first step of cellular respiration that occurs in the cytoplasm. Very good. How about this one? It is the process by which pyruvate is broken down in the presence of oxygen. Take note, in the presence of oxygen. What is that process? Correct. That process is called aerobic respiration. Very good. Due to the presence of oxygen. While anaerobic respiration is when there is no oxygen in the cell. How about this one? What do you call to the reaction that forms 6-carbon compound or citric acid? What is that reaction? The second stage of cellular respiration that occurs in the inner membrane of the mitochondria. So that stage or process is called Krebs cycle. Very good. How about this one? The final electron acceptor in the cellular respiration. What is the final electron acceptor in the cellular respiration? The correct answer is oxygen. So oxygen plays a great role as the final electron acceptor in the cellular respiration, especially in the electron transport chain, as it carries or it is the last molecule which receives the electron produced from the or released from the NADH and the FADH molecules. So, congratulations! You did it. You made it for the third module entitled The Cellular Energy. So, if you have any questions, suggestions, or clarifications, you are free to send us an email in the website online. And also, there are provided materials as exercise and examination. You can download it. You can download it in our site. So, uh, so once again, I am Dave. I am Teacher Dave, your junior mentor, and thank you for watching our module. Have a great day.